Inside a laboratory in Cambridge, England in 1977, a quiet revolution was beginning. A scientist named Fred Sanger was working with a molecule so impossibly small that you could never see it with your eyes, yet he was determined to read its complete sequence. He was trying to answer a question that had seemed utterly impossible just years before. Could we actually determine the exact order of the chemical bases in DNA? Could we read the genetic code letter by letter? What Sanger didn't know was that he was about to create the foundational technology that would eventually unlock the entire human genome and transform medicine forever. For decades, scientists knew that DNA contained information encoded in the sequence of its 4 bases: adenine, guanine, cytosine, and thymine. They knew that this sequence determined everything about an organism. But actually reading that sequence? It seemed like trying to count individual grains of sand on a beach while blindfolded. The DNA molecule was too small. There was no way to distinguish one base from another. It was the ultimate puzzle, and Sanger was obsessed with solving it. What made Sanger unique was that he didn't think like most biologists. He had won a Nobel Prize for protein sequencing, a technique that seemed obscure and impractical to most scientists. But Sanger understood something fundamental. If you could sequence proteins, you could sequence DNA. The logic was similar. The challenge was different, but not impossible. So he began experimenting with a method that involved creating chains of DNA with a controlled break at each position. By using a mixture of regular nucleotides and modified nucleotides that would terminate the chain, he could create a ladder of fragments, each one ending at a different base. Then, by separating these fragments by size using a process called electrophoresis, he could deduce the sequence. It was elegant. It was revolutionary. And when he published his method in 1977, most people in the scientific community barely noticed. But Sanger knew he had discovered something profound. He had created the first practical method for reading DNA, and within a few years, his technique, called Sanger sequencing, would become the standard method used in laboratories around the world. Sanger would win his second Nobel Prize for this achievement, the only person in history to win 2 Nobels in chemistry. The possibility haunted researchers through the 1980s. Sanger sequencing was powerful, but it was also tedious. To sequence DNA, you had to prepare it, run the reaction, separate the fragments, and then read them. One researcher might spend weeks sequencing a few thousand bases, but what if you wanted to sequence millions of bases? What if you wanted the complete genetic code of a human being? It would take hundreds of years and cost billions of dollars. The technology simply wasn't ready yet. Then, in 1990, something extraordinary happened. The US government announced the Human Genome Project. It was going to be the most ambitious biology project ever attempted. Scientists would work together across universities and countries to sequence the complete 3 billion letter human genome. Most people thought they were insane. The cost was estimated at $3 billion. The timeline was 15 years. The technology didn't even exist yet. But the ambition was irresistible. What followed was a race, not between countries, but between visions of the future. On one side was the official Human Genome Project, a public consortium working methodically and carefully. On the other side was Craig Venter, a brilliant, brash entrepreneur who founded a company called Genomics. Venter declared that he could sequence the entire human genome in just a few years using a technique called shotgun sequencing, where you break the DNA into millions of random fragments, sequence each fragment, and then use computers to assemble them back together like a jigsaw puzzle. The scientific establishment thought he was delusional. But something remarkable happened. Driven by competition, both teams accelerated their work. Scientists developed new sequencing. Machines. Chemists optimized the chemistry. Computer programmers wrote software to handle the staggering amount of data. By 2000, both teams announced that they had completed a draft of the human genome. Craig Venter had done in 6 years what people thought would take 15. The race had forced innovation. The technology had leaped forward. And when President Bill Clinton and British Prime Minister Tony Blair announced the completion of the Human Genome Project at a simultaneous press conference on June 26, 2000, the world changed. But the sequencing revolution was only beginning, because Sanger sequencing, the technology that had made the Human Genome Project possible, was about to become obsolete. In the early 2000s, researchers began developing what they called next-generation sequencing, or NGS. These new technologies worked on completely different principles than Sanger sequencing. One method, developed by a company called Illumina, used a technique called sequencing by synthesis. Imagine millions of copies of a DNA fragment attached to a surface. Then you flood that surface with fluorescently labeled nucleotides. When each nucleotide binds to the growing strand, it releases a flash of colored light. A camera captures the light. The computer records which base was added. Then you do it again and again, millions of times simultaneously on millions of different DNA fragments. You can sequence an entire human genome in a single run, and you can do it in hours. The implications were staggering. What had taken the entire Human Genome Project years to accomplish could now be done in a day or two. What had cost $3 billion could now be done for less than $1,000. The technology that had seemed like an impractical fantasy was becoming routine. And as the cost dropped and the speed increased, applications that had seemed theoretical suddenly became practical. Hospitals began using sequencing to diagnose rare genetic diseases. A child would be born with mysterious symptoms that confounded every doctor. The child would be shuttled from specialist to specialist. The parents would spend years in medical limbo, not knowing what was wrong, unable to treat it. Then, doctors would sequence the child's entire genome. Within weeks, they could identify the exact genetic mutation causing the symptoms. Suddenly, a disease that had seemed incurable had a diagnosis, and sometimes they could treat it. There was a boy in Minnesota who developed seizures so severe that no medication could control them. He underwent countless tests, brain scans, blood tests, neurological evaluations. Nothing helped. His family was desperate. Then, a doctor suggested whole genome sequencing. When the results came back, they found it, a single mutation in a gene called CDKL5. It was a rare genetic cause of epilepsy. The parents learned that the mutation was de novo, meaning it had occurred spontaneously in their child, not inherited from either parent. But more importantly, they learned what was causing the seizures. The family could pursue targeted therapies. They could connect with support groups for other families with CDKL5 mutations. The child had a diagnosis at last. In medicine, sometimes knowing what's wrong is the first step toward fixing it. These stories repeated across hospitals worldwide. Sequencing was solving medical mysteries that had mystified doctors for decades. But the revolution wasn't limited to diagnosing individual patients. Sequencing was about to transform our understanding of human disease itself. Cancer researchers began sequencing the DNA of tumors. What they discovered was shocking. Every cancer is genetically unique. Two people with the same type of cancer, say lung cancer or breast cancer, might have completely different mutations driving their disease. This meant that treating all lung cancers with the same chemotherapy drug was fundamentally wrong. A drug that worked beautifully for one patient might be useless for another, even if their tumors looked identical under a microscope. But if you sequenced the tumor, you could identify the specific mutations that were driving it. You could then find or design drugs that targeted those exact mutations. This approach, but if NGS had revolutionized medicine, it was about to revolutionize everything else too. In 2008, a company called Pacific Biosciences developed a sequencing technology that could read much longer stretches of DNA at once. Most sequencing technologies read in short chunks, maybe 100 to 300 bases. But PacBio could read fragments that were tens of thousands of bases long. Why does this matter? Because the human genome isn't just a simple string of letters. It has regions of repeated sequences, segments that are copied multiple times. With short-read sequencing, these regions are nearly impossible to assemble correctly. It's like trying to assemble a puzzle where multiple pieces look identical. But long reads can span across these repetitive regions and assemble them correctly. Then, in 2014, a British company called Oxford Nanopore developed something even more revolutionary, a sequencing technology so tiny and portable that it could fit in your hand. Nanopore sequencing works by passing a strand of DNA through a tiny pore in a membrane. As each base passes through the pore, it changes the electrical current in a measurable way. Different bases cause different changes. The sequencer detects these changes in real time and calls the base. No fluorescent labels, no cameras, no massive instruments, just electricity and a pore smaller than a nanometer. What made nanopore sequencing so revolutionary wasn't just the technology, it was the implications. For the first time, genetic sequencing didn't require a laboratory. A nanopore sequencer, no bigger than a USB stick, could be carried anywhere. A doctor could sequence a patient's DNA in a clinic. A researcher could sequence microbes in a rainforest. A field epidemiologist could sequence viruses during an outbreak. Sequencing had escaped the laboratory. This became abundantly clear in 2020 when the world faced something it had never encountered before, a global pandemic caused by a novel virus. SARS-CoV-2 appeared in China in December 2019 and spread across the world with terrifying speed. Hospitals were overwhelmed. Ventilators ran out. Death tolls climbed. And at the center of the emergency was an astonishing scientific response, powered by genetic sequencing technologies that had been developed just years before. Within weeks of the virus's discovery, Chinese scientists had sequenced the complete genome of SARS-CoV-2 and shared the sequence publicly. Within days, researchers around the world had analyzed the sequence and begun developing potential vaccines. The speed was unprecedented. A vaccine typically took years to develop, but with the complete Genetic sequence available, scientists could design vaccines in days. Moderna and Pfizer both designed mRNA vaccines based on the genetic code of the virus. These vaccines didn't inject a weakened or inactivated virus. They injected genetic instructions telling cells to make a protein that looks like the virus's spike protein. The immune system recognizes this protein and learns to fight it. The irony was profound. A pandemic that had been made possible by a virus's genetic code was being fought using genetic sequencing and genetic vaccines. The same technologies that had emerged from the Human Genome Project were now saving millions of lives. And throughout the pandemic, as the virus mutated, scientists were constantly sequencing new variants, tracking the virus's evolution in real time, updating vaccines to match the latest variants. Sequencing had become an essential tool for fighting infectious disease. But the revolution in sequencing was extending far beyond medicine. A group of researchers at UC Berkeley, led by Jay Shendre, developed a technique called DNA, sequencing for metagenomics. Instead of sequencing one organism's DNA, they sequenced everything in an environmental sample. Take a handful of soil, sequence all the DNA in it. You'll find thousands of bacterial species, fungi, viruses, all living together. You can identify species that have never been cultured in a laboratory, microbes that scientists didn't know existed. This opened an entire new frontier, understanding the microbial world that surrounds us and lives inside us. The microbiome, the trillions of microbes living in your gut, on your skin, in your mouth, turned out to be far more important to health than anyone had realized. Sequencing technology revealed that your microbiome is as unique as your fingerprint. The specific combination of bacterial species in your gut influences your immune system, your metabolism, your risk of obesity, even your mental health. Disease can be linked to microbiome disruption. Cancer patients who responded well to immunotherapy were found to have different microbiomes than those who didn't. Therapies designed to manipulate the microbiome are now in clinical trials. And then there was ancient DNA. The story of Neanderthals being sequenced had seemed miraculous, but as sequencing technology improved and became cheaper, scientists could now sequence DNA from fossils much older than Neanderthals. In 2013, researchers sequenced DNA from a finger bone found in a Siberian cave. The bone was 40,000 years old. The DNA showed it came from an unknown human ancestor that was more primitive than Neanderthals. They named it the Denisovans. We didn't know they existed until we found their DNA. A Danish scientist named S.K. Willerslev has spent the last 2 decades using sequencing to read the genomes of ancient humans. He has sequenced DNA from Viking tombs, from people who lived 10,000 years ago, from the earliest humans in the Americas. Each sequence is like a time capsule, a genetic snapshot of who lived and how they were related. He discovered that the first humans to arrive in the Americas came in multiple waves from Siberia, and that some of these ancestral populations have no living descendants. Others left descendants, and Willerslev can now trace modern Native Americans' genetic ancestry back thousands of years. This use of sequencing for archaeology and anthropology has fundamentally changed our understanding of human history. We can now do genetic genealogy at scale, tracing populations, migrations, and relationships through DNA instead of written records. A person can take a genetic test and discover they have relatives they never knew existed. They can learn what percentage of their ancestry is from different parts of the world. They can find their genetic cousins. But as sequencing became cheaper and more ubiquitous, new challenges emerged. The amount of data being generated was staggering. Sequencing one human genome produces about 200 gigabytes of data. Sequence 1 million genomes, and you have 200 exabytes of data. No human could possibly analyze this data manually. You would need computers. You would need artificial intelligence. This is where machine learning entered the story. Researchers began training neural networks to analyze sequencing data. A machine learning algorithm could look at sequencing reads and predict which bases had been called correctly and which might be errors. It could assemble fragmented sequences faster than traditional algorithms. It could identify disease-causing mutations from sequencing data with remarkable accuracy. And as more sequence data became available, the algorithms got better. Artificial intelligence and genetic sequencing had become partners. One company called DeepMind, owned by Google's parent company Alphabet, used machine learning to solve a decades-old problem in biology: protein structure prediction. They trained a neural network called AlphaFold on all known protein structures and amino acid sequences. Then they asked it, given an amino acid sequence, can you predict what 3-dimensional shape the protein will fold into? The AI's answer was so accurate that it was practically perfect. This transformed structural biology. Scientists no longer had to spend years crystallizing proteins and using X-ray diffraction to determine their structures. They could sequence the gene, predict the protein structure using AlphaFold, and understand what the protein does. The combination of sequencing and machine learning has created a feedback loop. Better sequencing generates more data, trains better AI. Better AI finds new insights in the data. Better insights suggest new sequencing experiments. This cycle is accelerating, and we're only at the beginning of what's possible. Today, sequencing costs have dropped so dramatically that they're approaching the cost of other routine medical tests. A decade ago, sequencing a human genome cost a few $1,000s. Today, it costs a few $100s. Within a decade, it might cost less than $50. At that price point, genetic sequencing will become routine. Every newborn could have their entire genome sequenced at birth. Every patient entering a hospital could have their DNA analyzed. Every cancer could be sequenced. The question won't be whether we have the technology to sequence. The question will be what we do with all the information we get. This creates ethical and social challenges that are profound. If you sequence a baby's entire genome at birth, you might find that they carry a mutation for a disease that won't appear until they're 40. Do you tell the parents? Do you tell the child when they're older? Do you use that information to influence medical surveillance or insurance rates? Some of these questions have legal answers, others don't. The technology has moved faster than society's ability to establish ethical frameworks around it. There's also the question of equity. Right now, most genetic data comes from people of European ancestry. Genetic research is skewed toward European populations. This means that medical treatments developed using this data might work differently for people of African, Asian, or other ancestries. There are calls to sequence the genomes of millions of people from diverse populations to create more representative databases. This is necessary for precision medicine to work fairly for everyone. Looking toward the future, sequencing technologies are becoming even more innovative. Researchers are developing in-cell sequencing, where the sequencing machinery works inside a living cell without extracting the DNA. They're developing multiplexed sequencing, where you can sequence millions of different DNA samples simultaneously in a single run. They're developing single-molecule sequencing in real time, where you can watch a single strand of DNA being synthesized and read the bases as they're added. One of the most ambitious projects underway is the Earth Biogenome Project. Scientists want to sequence the complete genomes of all eukaryotic species on Earth, millions of species. For the first time, we would have a comprehensive genetic catalog of life. We could understand how organisms are related at the molecular level. We could identify genes that are universal across life and genes that are unique to specific species. We could find genetic solutions to medical problems by studying the genomes of organisms that have solved similar problems through evolution. Another project, called the Genomes of the Human Microbiome, seeks to sequence the complete genomes of all microbes associated with the human body. We don't know how. Many microbial species live on us and in us. There could be thousands. Understanding their genetic diversity is essential for understanding how the microbiome influences health and disease, but perhaps the most transformative application of sequencing technology is one that barely existed a decade ago: direct-to-consumer genetic testing. Companies like 23andMe and Ancestry DNA have sequenced the DNA of tens of millions of people. Most of these people did it to learn about their ancestry or their genetic predispositions to disease, but this massive database of genetic information is now being used for medical research. Scientists can search the database to find genetic variants associated with disease. They can identify protective variants that keep people healthy even when they carry genetic risk factors. A woman in her 60s took a genetic test and learned that she carried a BRCA2 mutation, which meant she had a significantly increased risk of breast cancer. She consulted with her doctor and decided to have prophylactic surgery, removing her breast tissue before cancer could develop. She later learned that her genetic information in a research database had contributed to a study that identified a new breast cancer gene therapy. Her data had helped other women. This is the promise of large-scale genetic sequencing. Individual data contributes to collective knowledge that benefits everyone. Yet we must pause and acknowledge the dark side of this power. Genetic information can be used to discriminate. Insurance companies could use genetic data to deny coverage or raise premiums. Employers could use it to avoid hiring people with genetic predispositions to disease. Governments could use it for surveillance. China's mass sequencing of Uyghur populations has been accused of enabling ethnic profiling and oppression. The power of genetic sequencing to do good must be carefully balanced against its power to cause harm. There are scientists and ethicists working on these challenges right now. They're developing policies for genetic privacy. They're creating frameworks for the ethical use of sequencing data. They're advocating for laws that protect people from genetic discrimination. The technology itself is neutral. What matters is how we choose to use it. As we stand at this moment in history, the technology of DNA sequencing has fundamentally changed what's possible in medicine, in biology, in archaeology, and in our understanding of life itself. We can read the genetic code. We can identify diseases before symptoms appear. We can trace human ancestry across thousands of years. We can sequence viruses during pandemics in real time. We can discover new species in environmental samples. We can train artificial intelligence to make predictions that seemed impossible, and the technology is still accelerating. Nanopore sequencing gets longer reads with each generation. Illumina develops higher throughput instruments that sequence more DNA simultaneously. New technologies emerge from research laboratories with capabilities that would have seemed impossible just years ago. The cost continues to drop. The speed continues to increase. The applications continue to expand. In the coming decade, sequencing will become so routine and so inexpensive that it will transform into something more like a utility than a specialized procedure. Just as we routinely check blood pressure or blood glucose, we might routinely sequence DNA. Just as you can get your genome sequenced today for a few $100s, in 10 years it might cost $50. In 20 years, it might be free. When that happens, when genetic sequencing becomes as common as checking your email, we will have entered a fundamentally new era of medicine and biology, an era where every person's genetic code is known, an era where diseases are predicted before they manifest, an era where treatments are precisely tailored to individual genetics, an era where the invisible instructions that make you, you, are no longer a mystery. The revolution that began with Fred Sanger reading a few bases in 1977 has fundamentally transformed our relationship with the code of life itself, and we are only at the beginning